also 10 minutes is a good time to keep in terms of like mine is also, I think, around 20 something.
All right, we are live on YouTube.
All right, so we'll just do,
we'll do our usual wait for a couple of minutes and then get started since we have two just a bit after the hour. So Aashrith, how's Stanford? Are you enjoying California after all those years in Wisconsin?
Yeah, it's fun. I mean, I miss the snow, maybe for a couple of days, but it's definitely much better here in terms of weather and a lot of mountains and unlike the flat land. But yeah, I miss Madison too. I mean, that's almost like that. I was there for 6 years, and then that's almost my second home. So I miss that place. But California is fun to be in as well. But it's quite stressful how expensive it is, especially for a postdoc. I mean, I shouldn't say Boston is similar when you are in Boston, but yeah,
it's not as bad. Boston is more, I mean, it's rough, especially if you're in the city, although I lived outside the city for most of my time there. But yeah, I mean, the Bay Area is just, it's tough, right? So
yeah. But the weather is good. I'm happy about that. Yeah.
Good. I'm glad you're, I'm sure you're thriving in your new position. So
yeah.
I mean, the beginning's hard, right? Starting up a postdoc,
Definitely.
Switching areas, learning new stuff. It's always hard.
But it's getting better. I mean, things are getting more excited as you learn, and you can try out your own questions as once you know the system. So it's getting much better now, and it's also getting more excited. So exciting. So yeah.
Awesome. I'm so glad to hear that. Do you think we can expect Jacob to join us today?
I don't know. I didn't. I talked to him briefly, and he said that he may not show up, but I'm not sure. He may have a class or something. I'm not sure.
Okay. Well, I always like to have an excuse to see my old friend. And you're going to be presenting work from his lab, right?
From work from a PhD, yes.
Great.
I think we have hit 11, yeah.
Okay, yeah. And just checking to make sure. It looks like Zoom and YouTube are still both going okay. All right. So yeah, I think technical stuff, we are all set. So whenever you want to start, Ankita, I'll leave that to you.
Yes. Welcome everyone for another session of Cell Migration Seminar. Today, our first speaker is going to be Dr. Aashrith Saraswathibhatla. He received his bachelor's in mechanical engineering from Indian Institute of Technology, Gandhinagar in India. He did his PhD with Jacob Nottbaum at University of Wisconsin-Madison. I think he's going to talk majorly about his PhD work today. His PhD was in investigating various physical mechanisms that control collective cell migration in epithelial monolayers, which is one of Jen's favorite topic as well. So after his PhD, Aashrith has recently decided to do his postdoc in Chaudhuri Lab at Stanford. He is currently investigating the role of ECM viscoplasticity in mediating cell migration in 3D. So with this, I would really like to give the floor to Aashrith and welcome and thank you for presenting today.
Oh, thank you. Thanks a lot, Ankita, and thank you, Jen, for the invite. And I'm really excited to present in this great cell migration seminar series, and it's an honor to present in the seminar series. And thank you all for coming today to listen to my talk. Let me see if the presentation starts to work.
Yeah, looks good.
Awesome. And can you hear me fine? Okay. Sorry. Okay. My name is Aashrith. I'm currently a postdoctoral researcher at Stanford in the Chaudhuri Lab, but today I'll be presenting some of the work that I've done as a PhD at UW-Madison in Nottbaum research group. And the title of the talk is Coordinated Cell Deformations in an Epithelial Monolayer. So I'll get started with two videos over here. So what I have here is, I see a comment. Okay, sorry. So what I have here is a single epithelial cell that's migrating around. Let me play the video again. And we can see that the single epithelial cell is basically randomly moving around. But if I take the same cell, and if I just place it in a group, what we see is something very cool and very amazing. And we see a lot of large-scale order that's been developed. And basically every single cell here is nothing but a single cell that I have on the left. And we also see that cells are more persistently migrating, but unlike the single cell that's exhibiting more random-like motion. This is pretty good, but of course that also asks the question, like, what is the physiological relevance of such collective cell migration? And we are all familiar with the physiological relevance of it, but I would like to answer this question in a very specific cell migration mode way. And the way I would like to answer is basically looking into various modes of migration that cells exhibit in a cell monolayer or as a collective. So the first observation that I want to start with is rearrangements. So in a monolayer, cells rearrange. Here, what I have in this schematic is that the blue cells, which are not in contact, over time can become in contact. And it's a way of mode. It's a migration mode that cells exhibit in a monolayer. And in fact, such rearrangements can lead to very interesting physics. For example, here I have healthy cells and asthmatic cells, where asthmatic cells are shown to be more fluid-like compared to healthy cells, which are more solid-like. And in fact, some of my PhD work did look into the factors that control the cell shape and cell rearrangements in a monolayer. And in addition to the cell rearrangements, cells also migrate coherently as a group. They move as a group, they coordinate the motion as a group. And in fact, such coordinated motion is very critical in the Drosophila axis elongation. And some of my PhD work I have also looked into what controls the pack size in a coherently moving cell group in an epithelial monolayer. So here I have cells moving together, and the velocity vectors basically they are the instantaneous cell velocities, and the color defines the direction of the cell velocity. So you can see the groups that are moving together have similar velocity colors. And in addition to motion, cells are squishy. They also change their areas very significantly, even in fact their volumes, almost by 100 to 200% over their migration mode. And such cell area deformations are really important, basically in a case where the tissue is folding. So here I have a video where the highlighted cells are basically cells that are contracting their areas. And in this nice video, what the Martin's lab has shown is that cells can coordinate their area changes, and that leads to tissue folding. And today, I'll be basically presenting some of my work, looking into the physical mechanism that controls the coordinated cell area deformations in a monolayer. So I'll get started with that. So what we are interested in is basically how do cells coordinate their area deformations when they are in a sheet of epithelium. So the experimental system that we use is MDCK cells. These are kidney cells. These are very conventional epithelial cells that people use to study different developmental questions. And what I have here is cells confined in a 1 millimeter island. And we can clearly see that cells are, while moving around, they're also contracting a lot, and they're also expanding in areas. And specifically, if I just focus you all into these two circular areas, what I have here is in red, cells are expanding over time, while in blue, cells are contracting over time. They're becoming smaller. Let me play the video again. And in fact, some of the work from Angelini's lab, I have shown that the cell area can change almost by 200%. So the simple questions that we ask, or the questions that we ask is like, what are the physical factors that control the length scale of cell area deformations? Or in fact, to make it much simpler, what controls the size of these red and blue circles? Basically, the blue and red circles define the length scale at which cells coordinate the deformations. And in fact, such implications can be in development and disease. So with this, we come up with the hypothesis that the spatial length scale of contractile tension controls the length scale of this coordinated cell area deformations. Let me walk you through why we came up with such hypothesis. So in fact, there's a lot of literature saying that as cell area increases, the cell's contractile tension increases. Some of the early work from Tripad's lab have shown that the contractile tension on the y-axis over here is proportional to the change in area of a cell. And also, some of the work from my PhD advisor has shown that the stress rate of a cell is proportional to strain rate. So both of these imply that there's this active modulus for a cell where the cell area is proportional to the cell tension. And in fact, recently, there's a molecular mechanism that people have looked into, which is ERK-mediated mechanical feedback, where if you stretch a cell, the ERK activity gets activated in the cell, and that leads to cell contractility. And in fact, a cross correlation between ERK and area has shown a positive peak, saying that ERK is controlling the contractility and cell area relation. So with this ERK activity, what they also found was that the ERK activity is correlated in space, which basically suggests that there could be a possibility that cells could be coordinating a contractility in space, which is leading to this length scale of coordinated cell area deformations. So essentially, based on this literature observations, we hypothesized that the length scale at which cells coordinate their forces controls the length scale of the cell area deformations. So with this, we started to test the hypothesis. So before testing the hypothesis, we wanted to quantify the cell area deformations as well as the cell contractile tension. So for cell area deformations, if we look at this video, one way to go about that is basically segmenting the cells and getting the areas and getting the change in areas over time. But that's a very tedious process, and we all know that cell segmentation is a big challenge in the field. The way we approach this challenge is by just calculating the cell velocity vectors. And if we assume a cell that's decreasing in its area, we would expect the cell velocity field would be something like that's radially inward, right? And mathematically, if you take the continuum mechanics laws, a change in area rate, the rate at which the cell area is changing, is nothing but is equal to the gradient of the cell velocity. However, this has become much easier than just segmenting. You can just get the gradient of the cell velocity spatially and get the change in cell area rate. But the velocities that we calculate from DIC, or the image correlation, or PIV, they're very vulnerable to noises. So if you're taking a differential, the noises would basically amplify. And for this, the divergence theorem is currently for the rescue. And this is something that the Angelini's lab has shown, that if you just use divergence theorem, which is nothing but a area integral is nothing but the line integral of the velocity vector, here it becomes nothing but summation of the radial velocity over the area of the cell. So physically, what you can think is like the rate at which the cell area is changing is nothing but the summation of the radial velocities of the cells in that area of interest. So using this, we do calculate the divergence of cell velocity, which quantifies the cell area deformations. And positive divergence correspond to decreasing cell areas. And sorry, negative divergence correspond to decreasing cell areas, and positive divergence correspond to increasing cell areas. And qualitatively, we also confirmed that. So what I have here is two circles. In the blue, I have cells decreasing their areas, and in the red, I have cells increasing their areas. And we can clearly see that the divergence corresponds to the change in cell areas qualitatively here. And we also done quantitatively, and we have some of that in this paper. So now that we have measured the cell area deformations, the next thing is to quantify the length scale at which they coordinate the deformations, right? So for that, we use a simple metric called spatial correlation. In the spatial correlation, you're basically correlating the divergence at a spatial coordinate with divergence at different distances of different radii. Essentially, you're correlating how the divergence is correlated over space. And if there's no coordination at all, you would expect that the correlation plot would go to 0 within few microns, which is possibly a single cell length. But rather what we see here is that the correlation plot decays much slower, suggesting that there is a coordination between the cell area deformations, which is very visually evident over here. Now that we have measured the length scale at which cells coordinate the deformations, the next thing we want to do is to test the hypothesis. We want to measure the length scale of the contractile tension in the monolayer. And for this, we employ monolayer stress microscopy. And in this monolayer stress microscopy, what you do is you measure the tractions, and then you balance the contractile tension or the change in contractile tension by the tractions that cells exert on the substrate. So from this, you can get the intercellular stresses, and we define the contractile tension of a cell as the average of the normal stresses. In mechanics field, this is basically an invariant that you can measure, a scalar invariant that you can measure for a field of stress, and that's what we measure over here. So similar to the spatial correlations of divergence, to get a length scale of at which cells coordinate the tension, we do a spatial correlation of tension, and we see that the tension decays at a similar length scale compared to divergence, which basically is in agreement with our hypothesis that the contractile tension and divergence coordinate over similar length scales. So now that we have a way to measure the length scales of both tension and acyl area deformations, we started testing of the hypothesis. So we started to perturb the length scale of contractile tension and see how that affects the length scale of divergence. For that, the first target, of course, we thought of going with the obvious target, the E-cadherins. E-cadherins are basically that cell-cell adhesions that transfer the intercellular forces. So perturbing or basically disrupting the E-cadherins is expected to change the length scale of contractile tension in a monolayer. So that's what we do. We treat the cell monolayers with EGTA. So EGTA basically chelates calcium, which is necessary for E-cadherin activity. And what we see here is a clear, evident observation that cells move a lot and deform a lot in control. Compared to EGTA, cells hardly move, cells hardly coordinate any of the deformations. And to see if this EGTA has affected the length scale of contractile tension, we do measure that. We can clearly, qualitatively see here that the contractile tension has become much more heterogeneous in space, which suggests that there is a decrease in the length scale at which cells coordinate their tension. So now that we have seen this, to test the hypothesis, we looked into the length scale of divergence, and clearly from this video, it is evident that cells are decreasing their length scale of divergence, and we also measured that. And this is what I told about divergence being not correlated in space. We can clearly see that divergence is much more heterogeneous compared to a control case, and the length scale has decreased as well. So overall, in agreement with our hypothesis, disrupting the force transfer between any two cells disrupts the length scale of both tension and cell area deformations, and which also says that the length scale of tension might be controlling the length scale of cell area deformations. So in this experiment, we have decreased the length scale. Then we thought of designing another experiment where we increase the length scale of contractile tension, and this is where one of our recent observations comes into picture. What we have seen recently is that when you activate Rho using one of the Rho activator, there usually more stress fiber activity in those cells compared to control, which is a low serum. And we also see that cells align their tractions as a group. So if we just take the force equilibrium, a change in tension or the change in intercellular force is balanced by the cells or the traction that cells apply. So if you take stress, it is nothing but the integral of the traction, right? So if we take the reasoning that stress fibers generate tractions and tractions balance the change in intercellular forces, so increase in the alignment of stress fibers which generate tractions is expected to change the length scale of the contractile tension. So that's what we measure. In fact, that's what we see as well. We see an increase in the tension correlation length when you activate Rho, and which is quantified over here. So basically suggesting that multicellular traction alignment between cells can change the length scale of the contractile tension. So now that we have increased it, which is in contrast to the previous experiment, we see this has increased the length scale of divergence. And that's what we measure next. On the bottom, I have a low serum condition where cells are hardly moving or deforming. But on the right, I have a Rho activator condition. Let me play the videos again, where we see that cells are contracting as a group. And to quantify it, we also measure the divergence correlation length, and we clearly see an increase in the divergence correlation length when the Rho is activated. So overall, the data until now, what it points out to is that perturbing the force equilibrium in a cell monolayer as a way to change the length scale of contractile tension is controlling the length scale of the divergence. However, the chemical perturbations might be affecting a lot. We all know that if we perturb a cell monolayer or cells chemically, there might be a lot of side effects to it too. So we thought of investigating this hypothesis in terms of like physically perturbing the length scale of the contractile tension. Can we perturb it or not? So for that, we gave a reasoning that the island size, the size of the island to which the monolayer is confined may limit the length scale of the force transfer. So increasing the confinement of the island or the island's diameter may change the length scale of the contractile tension. And for that, we essentially confine these monolayers in different confinements, basically in a diameter of 500 microns, 1 millimeter, 1500 microns, and 3000 microns. And we see what we expect is that do we see an increase in the length scale of tension just because as the cells are increasing, or the size of the islands are increasing, cells can transfer these forces over longer distances. And in fact, in agreement with our reasoning, we do see an increase in the correlation length of the contractile tension as you increase the diameter of the confining islands. And in agreement with our hypothesis, we also see an increase in the divergence of the correlation length of the divergence, suggesting that even when you physically perturb the length scale of contractile tension, we still see an increase in the correlation length of the divergence, suggesting that the tension controls the length scale of divergence. So overall, let me demonstrate all the data that we have until now. What we have done is basically, can we perturb the force equilibrium, or can we perturb the length scale of contractile tension in a cell monolayer, and does that affect the length scale of the cellular coordinated cell deformations? And that's what we see here. Clearly, there's a physical, there's a causal effect of tension correlation length affecting the divergence correlation length. However, if we look closely at our hypothesis, there is an assumption that we still use in our hypothesis that the cell area is proportional to its contractile tension. So although cells are coordinating the tension, but if the relation between cell area and tension is not applicable, then we may not be able to see this coordinated cell area deformations. So that's where we started to test what is the role of this ERK-mediated cell area tension relation in controlling the length scale of the divergence. For that, we inhibit ERK kinases using U0126. And what we see here is that with ERK inhibition, so this is just an image from the literature where the ERK activity is correlated. But when you inhibit the ERK, we see a decrease in average contractile tension in a cell monolayer, which is expected just because ERK is controlling the contractility of a cell. But we don't see a change in the spatial correlation of contractile tension, which is opposite to what we expected. And moreover, in contrast to our hypothesis, there's no change in the length scale of the contractile tension, but there is a change in the length scale of the divergence, which is opposite to what we hypothesized, which clearly suggests that ERK inhibition basically ERK inhibition is affecting this length scale of divergence. So to further quantitatively verify this, we see if ERK is actually affecting the relation between area or not. We started to measure the single cells tension and single cell area so that we can do a cross correlation between that. For that, we developed this methodology where we track individual cells over space and time. So you basically have the spatiotemporal values of every cell's coordinates over space and time. And from this, you can basically approximate every cell as a polygon, which gives the cellular areas. And you can also overlay the monolayer tension on this Werner tessellation, which gives the individual cell contractile tension. And from this, to see if there's a direct relation between cell area and tension, we did temporal cross correlation. Essentially, this is similar to spatial cross correlation, but except you're doing it in time, and you're also doing it between two different signals, which is area and tension over here. And what we see here is a clear positive peak at 0, which suggests that the cell area is proportional to cell tension. And when you inhibit ERK, which is expected to diminish this relation, that is what we see when you keep on increasing the concentration of ERK inhibitor. We see a decrease in the peak as well as the red line, which is 40 micromolar ERK inhibitor. It completely disrupts the relation between cell area and tension. So with these findings, what we propose, a physical mechanism for coordinated cell area deformations, is that there's a force equilibrium between the cells that basically makes the cells coordinate the tension. If one cell is contracting, the other cell has to balance that force, which makes the neighboring cell contracting as well. In addition to this, there is this temporal coordination of cell area with its tension that is mediated by ERK. So there's a spatial coordination of tension and a temporal coordination of tension, which gives us to this coordinated cell area deformations. With this, I'd like to acknowledge my PhD advisor, Jacob, and the co-author on the paper, Jay, and the research funding. And I'd like to thank the cell migration organizers, Jen and Ankita, and thanks a lot for the invite. And thank you all for being here. I'll be happy to take any questions.
Thank you so much, Aashrith. While people are popping more questions, there is a question from Katie asking, have you thought about disrupting alpha-catenin to test effects of maintaining cell-cell adhesion, but disrupting the force sensing?
That's a very good question. We tried, sorry. We did try to acquire the cells. We couldn't get the cells that where we thought of inhibiting the alpha-catenin, but we did not do that experiment. But I think even that experiment is expected to give a similar result as EGTA, because you're essentially not letting the cells transfer the forces by disrupting the alpha-catenin, which links actin and ecadherin. That's what my guess would be.
Great, thanks. One thing that I was wondering is I would expect that all of your calculations would be strongly affected as cells are changing area and changing density due to cell division. So
Right.
Was that something that, did you just look on a short enough time scale that it didn't matter, or did you inhibit division, or was it not really an issue?
The cell density is definitely a big issue because of the cell division. So we did look at the timescales where we looked at the shorter timescales where the cell density doesn't really drastically change. But I do agree that if you change the cell density by just letting cells proliferate, the length scale might be affected by that. But we were careful about that.
Yeah. No, just because you're right, you have this calculation that looks at sort of the flow of area changes.
Right.
So it could be biased.
And the divisions are quite random in location. So even if there are divisions, I mean, I'm sure there are divisions happening, the velocity field might be very localized for such divisions. I know like some of the work from Ladou's group have looked into such divisions and cell velocity fields. But I wouldn't expect that such divisions would affect the divergence that we calculated over here.
Yeah, great. Okay, thanks a lot. I appreciate it. We have the next question from Ernest Liu. Thanks everybody for pouring these into the chat. Ernest is asking, does the spatial activity of ERK correlate well with cell area or contractile tension?
It does correlate well with the cell area changes, clearly inhibiting. We haven't really looked into the ERK fluorescent activity. We didn't have the cells for it, and I don't think we had the expertise to look into such FRET activity. But in fact, like just inhibiting the ERK itself changes the length scale of the cell area deformations. As you can see in this video, that cells have just basically become static without really changing their areas. And what we have done, something that hasn't been done in the literature, is that just to quantitatively look at the relation between the area and tension of a cell in the presence of an ERK inhibitor. And that's what we see when you inhibit the ERK. You can clearly see a gradual decrease in this area tension relation, which strongly supports that ERK is controlling the cell area and tension relation.
Awesome. Thank you. That's fascinating. Next question is from Curtis. He has two questions, so I'll go one by one. Says, awesome talk. Did any of the perturbations you looked at induce a time lag between the area and the tension?
That's a very good question. I guess, I mean, if I want to rephrase the question, is maybe it's looking into the viscoelasticity between the area and tension, like kind of looking at deformations. And we did not, to answer your question, we did not see any such time lags. And I'm not sure if we should expect to see a time lag with ERK or not, because it seems like a lot of research, even work from Stanford Lab and of course from some of my advisors, it seems like there's a linear elastic relation between cell area and tension. And I don't know if I expect a time lag between tension and area or not.
Awesome. He has a second question asking, did you look at the RMS velocities? And if so, what are the relationship between various correlation lengths and RMS velocities?
That's a very good question. I don't have the data right now, but we do have that in our manuscript or in our paper that RMS velocities do decrease whenever the cells don't coordinate their deformation. Somehow there's a correlation between the cell velocity and the rate at which they change their areas. Yeah, they do change. They do decrease in all the cases or they do decrease or increase whenever there's a change in the length scale in the divergence.
Fantastic. Our next question is from Sang-Yoon Han, asking, what do you think is the upstream regulator for both tension and cell area? Do you think the tension itself can act as an input as a feedback for area regulation?
That's a very good question. Let me try to think about that, because I don't know what if you're talking about the upstream cell signaling factors. I can think of row activity and everything, but in terms of cell tension and area feedback, it's a very interesting relation to me. At least I find it really cool in the sense that the tension that we calculate, let me just go through this, the free body diagram over here. The tension that we calculate, it's a combination of passive tension that cells might be experiencing as a solid body in addition to its own active tension. So I'm sure there is a feedback, at least the correlations that we see, but I'm not sure which part of the, how is cell area is related to active tension and passive tension in a cell. Because when you're stretching a cell, of course, there's this passive tension just because you're inducing a deformation on it. But cells also actively change the tension when you put a passive tension on it. So there is a relation. I'm not sure what the exact relation is, but it will be an interesting experiment just to decouple the role of passive and active tension in a cell and how is that related to cell area in general.
Awesome. Next question is from Ajay Chitnis, who's also our speaker in this season, is asking, how well does Voronoi analysis compare with the actual cell boundaries?
That's a very good question. And that's a question that one of the reviewers asked us during
maybe it was him.
So it does well. So it really depends on the cell density. So most of our experiments we perform that medium cell densities, which are to be specific about that, I don't know if that makes sense or not, but it's around 22,000 cells per millimeter square. At such densities, the cell areas do correlate, or the Voronoi approximated cell areas do correlate well with the actual cell areas, and we also have some of this data in our supplementals. But if you decrease the cell density, like very sparsely located cells, of course, the cell areas are not that correlated with the Voronoi tessellation. But as you keep on increasing the density, where the cells are just become polygonal at very high densities, they correlate much better at such high densities. So it really depends on the density of the cells. But what we have seen is that these approximations, at least the cell area approximations that we have done and all the quantities that we have computed based on these approximations, correlate well even with actual cellular outlines. So at least in our data, it correlates well.
Cool, thanks. I mean,
we lost you, Ajay. I think it's for everyone. Hello?
Yeah, I think.
Uh-oh. We can't hear
No,
you.
we can't hear you, but we actually do need to move on to Ankita's talk. So I'm going to ask you, but please, if you two want to continue this conversation afterwards, but I just want to ask one last question from Yuhan Zhao, asking, apart from EGTA, have you tried any ECadherin blocking antibodies?
Blocking antibody has always been a challenge. I've tried DECMA, which is basically this antibody or functional antibody for Cadenin, but I've never got that working for some reason. But that would be an interesting experiment to do. But I think to give another, I don't know, some of my experience with antibodies is that it really depends when you add it. If you add it when the monolayer is by itself, maybe the antibodies are not really getting through the monolayer just because there are tight junctions that are blocking it. but you may have to add it like when you trypsinize them and then add it, and then you make a monolayer out of it. So I think there's a big challenge over there, how to use the antibodies exactly to make that work in the first place.
All right, fantastic. I appreciate all the questions. Thank you, everybody. Thank you, Aashrith,
Thank
for
you.
a
Thanks for the
fantastic
questions,
presentation. And we can always continue this conversation afterwards. For now, I am going to ask Ankita to go ahead and share her slides while I introduce her. So I am so happy to be introducing my colleague and friend, Dr. Ankita Jha. Of course, you all know her from this seminar series. The beautiful images that you get every week in your inbox and that you see on Twitter are the posters are put together by her. So we can also take a second to thank her for all of her efforts in making the seminar series happen. So for the introduction, Ankita did her undergrad at the Indian Institute of Science Education and Research in Pune in India. Then she moved to Marseille in France to do her PhD with Professor Thomas Lequi. And there she worked on quantitative control of GPCR's organization and signaling in epithelial morphogenesis using Drosophila embryos as a model system. And she's now currently a postdoc in Dr. Claire Waterman's lab at the NIH. Her interests lie in understanding the plasma membrane organization and signaling during mechanical processes like cell migration. And she's currently working on receptor organization and polarity during bleb-based migration. And that's the story that we're going to hear about today. So without any further ado, I'll hand it over to you, Ankita, and welcome.
Thank you so much. I really want to take this time to thank Jen, Adam, and Bex, who actually started the seminar. I got to do, the easy job to come in later on when everything was established. And I almost feel emotional presenting in the seminar series because this is something so close to our hearts. And as Jen said, not only this has given us like awesome colleagues, but also friends. But, during COVID time, nobody thought it would happen. So I'm just going to use my privilege today and present my work here. And I'm really looking forward to get some inputs from you all, because I think this is going to be, this is such a perfect crowd for this sort of work. And I'm going to switch gears from really amazing work, which Aashrith showed about collective cell migration. And I'm going to be talking about single cell migration and more specifically, bleb-based migration. So as Jen mentioned, I am a postdoc in Claire Waterman's lab. And something which I'm going to talk today is about how cells often move through really confined spaces. And I usually really like to start my presentation with this really beautiful movie from Eric Sahai's lab, where you see these melanoma cells migrating in vivo by throwing out a lot of these huge blebs. So what are these blebs? Blebs are often classically defined as pressure-based protrusions, which are formed because you have a cortex rupture or a leakage from the cortex that leads to formation of these small protrusions, which are often filled with cytoplasm. So they also expand because you have contractility around the neck, which is constantly squeezing and leads to the expansion of blebs. So you can think it of like a balloon which you squeeze from one side and the other side is constantly protruding. At some point, the cortex sort of reforms around and retracts that bleb. So that sort of is what classically blebs are thought to be. But we know that cells often have to move through really sort of extreme environments when they're migrating in vivo. And one such extreme environment is actually confinement. So that has been also measured in vivo, that these confined spaces can be as small as even 2 to 3 microns. So if you again look at these melanoma cells, these cells have to really squish through these small pore-like space here in the collagen in order to move forward. So this was something really fascinating to me. And in order to understand how cells move through confined spaces, we can try to recapitulate that in lab by putting cells under vertical confinement. So what you see here, we put cells in a dish, we coat the surface for low adhesive environment. So I'm not going to be talking about any adhesion-based migration. So I apologize to anybody who's interested in integrin signaling. And what happens when we confine this, so this is going to be a 3 micron confinement, is really fascinating because you see these cells starts to throw out a lot of these bleb-like protrusions. But at some point, the cell is going to make a decision to make one really giant bleb and migrate in a very persistent fashion in the direction of formation of that bleb. So again, going back, looking at that really giant bleb moving forward, you will see that it's really migrating where the bleb is formed and is just sort of carrying the cell body as a trailing behind. And this work was really well done by Matthew Peel's lab and Verena Ruprecht, but also by Jeremy Lowe, who now has an independent lab of his own, but at that time was working in Claire's lab. So how are these blebs moving forward? This really giant bleb is often powered by fast actin retrograde flow. So you're looking at actin here, and you can see that how actin is moving from the tip to the base of the bleb. So I sort of like chopped out the cell body here. And just to remind you, and this is the direction of the migration. And this was really fascinating to me, not only that this was showing a really fast actin retrograde flow, but the actin architecture was so different from what we know from a classically lamellipodia-based migration, right? So if you look at the bleb tip here, you actually see more bundle actin, whereas the back, where there is a lot more myosin that maintains the contractility at the back of the bleb, you see a more mesh-like actin. So when I came to Claire's lab, I was really fascinated by these structures itself because they're maintaining sort of a front and rear without any external chemical cue that would lead to this sort of maintenance of polarity and would sort of migrate in a very persistent fashion. So I sort of decided to think about if any kind of receptor signaling was required for this bleb stability, and really thinking about what we know from adhesion-based systems. So in adhesion-based migration system, we know that the front is really established because of the ECM engagement and receptor signaling, more often integrin signaling that leads to the stabilization of focal adhesions also. And that also leads to PIP3 and RAC in the front that regulates a branched actin sort of behavior in the front of where the lamellipodia is formed. And this is, again, a very simplistic way of describing what goes on in an adhesion-based system. However, we know the back of the cell often has high levels of PIP2 and Rho-A GTP signaling that leads to maintenance of contractility at the back, which is, again, like pushing it forward. And the kind of actin architecture you have is actually bundle actin at the back. So really, to compare with the system, I started asking questions, is any kind of receptor signaling required for bleb-based stability? And if so, is it really polarized? And if the receptor signaling is actually polarized, what would be the mechanism that control this asymmetry? So I sort of went back again in the melanoma system, which was really well established in the lab with Jeremy. So what Jeremy had shown that in A375M2, ERK signaling was really confined in these huge blebs and was responsible for maintaining actin architecture. So I decided to sort of go upstream of that signaling. And in the absence of integrin signaling, what becomes really important is receptor tyrosine kinase signaling. So I decided to target EGFR. So EGFR signaling often can activate a PI3K kinase, which leads to more PIP3 activity, and can also activate RAC and RAS GTP that eventually leads to more ERK signaling in a system. So what happens when you inhibit ERK signaling in the system? So if you look at the cells on the right here, these cells would throw out a bleb, but fail to maintain it. And this is what is analyzed over here. So if you look at a control system, which is in gray over here, the blebs can be maintained as long as even an hour. However, inhibiting EGFR led to sort of throwing out the bleb, but it would retract back very quickly. So that showed that RTK signaling was really important to maintain bleb stability. So here is one target which I found. Next thing which I really wanted to address was, where is this EGFR signaling really going on? So if you just look at EGFR, what was really fascinating to me that it started to show these sort of gradients where you have more EGFR at the back of the bleb. And this was sort of counterintuitive because when we think about the receptor signaling, you always think that that is in the front of the cell rather than at the back of the cell. So I decided to now go for actually looking more downstream about the polarity markers that we know in our system, which is RAC and Rho. So just looking at the RAC sensor, you can clearly see that the RAC actually marks the back of the bleb here, which was, again, a reverse of what we know in an adhesion-based system where RAC is often in the front. if you look at the rho signaling, it was completely reversed. So now actually rho GTP is in the front. So you see the rho sensor more in these bleb tips. So this was really fascinating. There is some sort of a polarity signature, but it's entirely reverse of what we know in an adhesion-based migration system. So the next thing I really wanted to show was whether this sort of asymmetry in the signaling is really required for bleb stability or not. Sure, they are asymmetric in nature, but is it really important for bleb-based migration? So what I decided is to activate RAC in the tip of the blebs where it's absent. So just remember, RAC is actually in the back of the bleb. So I sort of mislocalized RAC activity in this experiment. So this is just, again, a control where you're looking at F-tractin, and you can clearly see these bundles going from the tip to the base beautifully. And the bleb is actually very dynamic in nature where you will see it grow. However, once I activate RAC by using these optogenetic tool from Klaus Hahn's lab, you can clearly see initially it's doing okay, but at some point you will see this sort of bundle disappear, which is sort of what is shown here, that the actin architecture changes massively. But not just that, if you look at the length of the bleb for a lot of these blebs, it eventually starts to sort of go down. So that shows that this, if you mislocalize this RAC activity, it's really important. In general, RAC activity on the back is really important for this bleb stability. So I'm not going to show you, but if you actually activate EGFR in the tip, which again, EGFR is not present at the tip of the bleb, you kind of see a very similar phenotype. So that showed that these gradients are really important for bleb stability in the system. So the next question I sort of wanted to ask was, how are these asymmetries, like what are the mechanisms of this asymmetry in the system? And really thinking about asymmetry of these transmembrane receptors, and particularly EGFR in my case. So one of the hypotheses which I could quickly sort of ruled out is that it's not happening because of any sort of bulk plasma membrane flow from the tip to the back, because if you look at the proteins like GPI, which is on the outer leaflet of the plasma membrane, or something simple as CAX, which is on the inner leaflet of the plasma membrane, there is actually no gradient. So there is no sort of a plasma membrane flow going on from the tip to the back of the bleb. The next thing we wanted to check was whether this was happening because of any kind of very specific endocytosis or exocytosis. And this is a harder question to tackle, because if you block endocytosis or exocytosis in systems like this, it's a very hammer-like experiment. So what I decided to do was to bleach the whole bleb and look at the recovery. And something really fascinating, which I found out when I look at the recovery in these blebs, and these are individual cells, most of the recovery really happens just around the neck area. It doesn't really happen in the tip area. This tells us two things. First, there is no specific exocytosis going on anywhere. It kind of looks like all this recovery is happening around the neck area. The second thing which we could also see was that the recovery was still in a form of a gradient. So imagine a transmembrane protein which could, sort of move around or diffuse around in a very freely manner would have been able to make this gradient flat if it was not somehow restricted at the back. So a third very sort of important hypothesis which I wanted to test and was kind of close to my heart was whether this was happening because of the advection. So again, to remind you, there is a fast actin retrograde flow from tip to the base. So one of the hypotheses could be that all this is getting dragged by that actin. So I decided to look at the EGFR recovery rate and this close to the gradient, and see if we could see any sort of a directional flow. And this was a slightly tricky experiment because at some point you will get diffusion from both sides. So it's just the initial time points you have to track. So by tracking it, what was really fascinating to me was that actually the recovery was more directional towards the tip of the bleb rather than the back of the bleb as I would have hypothesized. So this really showed that it's actually not a retrograde flow, but rather an anterograde flow, which was again a counterintuitive for us to think that there is so much actin which is moving in this fashion, but it's not really bringing those receptors to the back of the bleb. So there is some other mechanism which is doing it. So we decided to bring in Professor Jason Hogg and his student at that time, Ankit, to sort of start thinking about this system, and if we could really model and think about what other parameters could be important. And one of the key assumptions of this model was that all the proteins are initially made in the cell body. And this is a very valid assumption because these blebs are protruding out from the cell body. And also most of the cellular organelles are still in the cell body. So there is some sort of a major flux of these transmembrane proteins which are going on from the cell body towards the bleb. And what Jason and Anka decided to subject these transmembrane proteins to was lateral diffusion, some sort of bulk flow or flux, which I just talked about, and also the protein turnover, which is going on more at the back of the bleb, which came out of my FRAP experiments. So something which was really fascinating that came out of the model at the same time when I was doing experiments was the membrane flow. So what Jason now showed from this model that the membrane flow is rather entrograde in fashion, which I also showed from my experiments, which I was unable to explain. Another major thing which came out from this model was two important factors that could regulate sort of the shape of these gradients. So something what Jason and Ankit started calling was this local corralling or sieving coefficient. So this is when these transmembrane proteins are in interaction with the underlying actin cortex, that can definitely change the shape of the gradient. So it's not the retrograde flow, but some sort of interaction with the underlying actin cortex that can change the shape of the gradient. And the other important thing was the membrane turnover. So this sort of gave me an idea that, oh, probably there is something else which is leading to, the sort of gradients in the blebs. Another piece of evidence that shows that it's not retrograde flow is by looking at this membrane proximal actin sensor. So this came out around in 2020 by Tobias Mayer's lab, where there is the sensor which has CAX domain and an F-actin binding domain, but it only labels actin, which is very close to the plasma membrane. And again, to remind you, there is a lot more F-actin if you just look at F-actin overall. But if you look at the sensor, the tip of the bleb is actually like a classical bleb where you don't have that much actin close to the plasma membrane. So that you can imagine that the retrograde sort of flow will fail now to bring all those proteins to the back. So that supports the hypothesis that it's not because of the retrograde flow of these transmembrane proteins, but there is some other mechanism that leads to these gradients. So, okay, this was really fascinating. I decided to sort of think back about how these gradients are established over time. So if you look at just EGFR gradient progression over time in a single bleb, you will see that the gradients are established very early on. That led me to think that there probably is a mechanism to restrict diffusion of these transmembrane proteins at the back of the bleb and just is like keeping it at the back and not letting it go forward. I quickly wanted to test if there was some sort of restricted diffusion going on at the back of the bleb. And this can be done by a simple FRAP experiment where if you look at the spot FRAP for EGFR at the base of the bleb, it's actually lesser. So that means that EGFR is actually restricted at the back of the bleb rather than in the tip of the bleb. So this was really fascinating, which got me back to the picket fence corralling model, which was given by Kusumi and Fujiwara. And this is a model sort of very well established to think about the membrane organization in general. So if you think about these transmembrane proteins, which are often free to sort of diffuse around, this protein can be corralled or can be restricted by the presence of these other huge transmembrane proteins, which are referred to as picket proteins, that are relatively immobile. So they're not free to move around. So what these picket proteins do is that they can interact with the underlying actin cortex by proteins like ezrin, rhodixin, and myosin, and can really like sort of latch on and restrict the mobility of other transmembrane proteins as well. So thinking about what picket proteins could be important in the system, it was sort of no-brainer to think CD44 could be important. And this is because of multiple reasons. So CD44 is a picket protein, but is also a glycoprotein very established in the field of cancer because it can interact with MMPs, can also act as core receptor for a bunch of other receptors. And another important thing for CD44, especially in the field of melanoma, is that it can interact with hyaluron, which is highly present in your skin. So if you remember the creams you put has a lot of hyaluronic acid. That's because it's to supplement your skin with it can sort of grasp a lot of water and stuff. So I quickly wanted to check whether CD44 was really in a form of a gradient in the system. So if you look at the immunostaining here, you can actually see CD44 forms a very strong gradient as well. And also proteins like ezrin, which would lead to these Picket proteins and will latch these Picket proteins to the underlying actin cortex, was also present in huge amounts in the bleb. So if you look at just ezrin, it's fascinating that there's so much ezrin in the bleb, but not so much in the cell body. So this was really cool to me, and something I want to now establish was whether CD44 was really important to establish or corral these EGFR gradients. So I quickly wanted to test whether CD44 was actually important in the bleb stability or not. There's something which I see is very similar to EGFR inhibition. So you can clearly see, again, these cells are able to throw out blebs, but they're not stable. They get retracted back very quickly. So that showed CD44 was indeed important in this system. And the next thing was to actually check the gradients itself. So again, to remind you, EGFR has a very strong gradient in the blebs where you see a lot of EGFR at the back. But the moment I get rid of CD44, you start to see that now the blebs actually have sort of a more flat gradient. So it's not that sharp anymore, which is what is shown over here. So this was really interesting because clearly CD44 is required not only for the bleb stability, but also for maintaining this gradient, which we know by my optogenetic experiments that is important for the bleb stability. Another thing now I wanted to test was whether CD44 was indeed corralling EGFR. So again, if I do a spot frap for EGFR at the back of the bleb and now compare it with once I remove CD44, and you will again see that the gradient sort of disappears, but at the same time, the T half of the EGFR goes down. That means now EGFR is free to move around. So it's moving much faster. The diffusion is much faster. So now sort of the model I have for the establishment of French rare polarity is that the back of the bleb is really established by this, high levels of CD44. So there is more CD44 that can corral other receptors like EGFR. Now, because we have more EGFR at the back, that leads to higher PIP3 signaling, more RAC signaling, which I showed you by the sensors over there. And because we have more actin closer to the plasma membrane at the back of the bleb, this whole system of corralling and picketing works really well. So everything sort of is established because of the corralling at the back of the bleb. However, now in the front of the bleb, you have less CD44. So whatever receptors now manage to go to the tip, which is very less in general, are not able to signal as much. That leads to sort of higher levels of PIP2. So you just have high levels of PIP2 that supports more Rho-GTP activity at the tip of the bleb that would lead to more bundled actin. And also something we know that the actin is sort of far from the plasma membrane. So it's again goes against this idea of like latching onto the underlying actin cortex. So this is how the polarity is sort of established in the system. So now I quickly want to bring you back to the adhesion-based migration and comparing bleb-based migration system, because it's really fascinating that both of these systems are using the similar sort of components, but completely in an opposite fashion, where PEP3 and RAG-GTP controls branch actin at the tip, or not tip, at the front of the cells in adhesion-based migration. Whereas in bleb-based migration, you have higher levels of PEP2 and Rho-GTP signaling that leads to more bundled actin, because that we think is probably controlling something like formin to lead to more bundled actin at the tip. However, the back really, However, the back at the adhesion-based system has more Rho-GTP, but the bleb-based has more RAG-GTP going on, and that leads to more, sort of actin, change in the actin architecture at the back of the bleb. So some of the things which now I can ask in the future is, what are the molecular mechanisms for Rho-GTP activity in the tip? Because you can imagine now that there are not many receptors there. And one of the hypotheses I have that the activation actually happens at the back, and there is probably some mechanism for these Rho-GEFs to move at the tip. And then the whole antagonism mechanism about rho and rack, which could probably sort of maintain this polarity. The other important question is to ask, how are these polarity components assembled when cells often switch from one mode to another mode of migration? And it will be really fascinating to now think about the temporal regulation of this receptor organization, because receptors are often in the front in this system and at the back in the system. And one of my long-term goal is to also understand what is the impact of the changing extracellular matrix. And I can pick something like melanoma disease progression, where we know that MMPs and hyaluronic acid actually changes. And we do not really understand the relation between that with respect to CD44 signaling and any kind of these modes of migration. So with this, I really want to come to my acknowledgement and really want to thank Claire to sort of give me the freedom of things I want to do in the lab, but also such amazing support from my lab members and throughout my project. I would also really like to thank my collaborators, Jason and Ankit. And I did not talk about the work I did with Abhishek and Anthony. I also want to really thank you for listening my talk, and I will take any questions and suggestions you might have. Thank you so much.
Wonderful. Thanks so much. So before we dive into the questions, I will just say, if any of you don't want to hear me talk this entire time, feel free to use the raise hand function, ask questions yourself. Otherwise, I'll just go through things in order. So first, we have a couple of sort of technical questions from Nada Albaba asking about, can you talk about the confinement apparatus and how you're passivating the plate to ensure the confinement? And then they're also wondering, how do you technically, how do you immunostain cells that are undergoing this bleb-based migration, and are you fixing them while they're in confinement? So I think just a little bit of more clarity about the system you're using.
Absolutely. Yeah. So this, the vertical confinement system is something which was developed in Matthew Peel's lab. So it actually has these PDMS pillars, which are of definite size. So you crush it and you sort of have those pillars, which will maintain the size. Having said that, you can quickly test it by using beads. That's what we initially did to make sure that the confinement was really 3 microns. So you can use a 3 micron bead and see if they're not sort of jiggling around. The immunostaining is really painful. So you have to do it under agarose, again with beads. And often it's very tricky because these cells are not adhered to the substrate. So I actually often coat them with either BSA or PLL PEG. That would lead to not, like, for these cells to not form any focal adhesions. So you have to really slowly, so really contact me for the immunostaining protocol, but I can tell you it's, again, very, very painful.
Thank you. Yeah, when you see such amazing imaging and just all of this incredible work, it's sort of stunning to us experimentalists thinking, how the heck did you do all that? So we have a suggestion from Olivier Pertz suggesting to look at EGFR activity, and they've put a link in the chat with some.
That's absolutely something which I'm doing in process. So I actually have looked at EGFR activity by few methods which I didn't talk about. There's one which is by fluorescence anisotropy, is to look at whether EGFR is more clustered. So if they're more clustered, their activity is more. So I completely removed that. That's a wonderful suggestion because I know Jared Potticher has a new EGFR sensor, which I'm going to try out. So this is in process. Thank you so much.
That's the link that he sent. So it looks like you all are on the same page.
Yeah, I'm in contact with him. I have been using his optogenetic tools actually.
Fantastic. We have a question from Taehyung Kim saying, in the tip of the bleb, the increased actin bundle seems more straight than branched. So do you think the front side of the bleb is more Foreman mediated and the back of the bleb is more ARC2/3 mediated for the branched actin there?
Yeah, so in terms of the Foreman mediated, absolutely, like that's what our thinking is, especially looking at the actin architecture. I personally have not explored it too much, but that's where our thinking goes because of the higher row signaling as well. About ARC2/3 signaling, it's a little bit tricky because at the back of the bleb, if you just do CK66, blebs do not really react to it very quickly. Like it takes them a while to sort of, for the whole system to sort of collapse. But I don't know whether that's happening because now you are, sort of doing something major to the cell body as well. So I can't really answer whether it's up to 3 mediated or not. If you look at the up to 3 localization itself, there is some tiny specks you kind of get at the back of the bleb and more in the cell body, but I actually don't know. That sort of is the prediction, but I do not have experiments to really say that that's true or not.
Great, thanks so much. We have a couple of questions from YouTube, so I'm going to switch over to that. From Nagaraj saying, great talk, and a couple of questions. First, are there any microtubules seen in the blebs, or is it exclusively actin driven?
So it's a wonderful question. There's actually very, very beautifully like long microtubules that go all the way. So something which doesn't happen is they don't reach the exact tip of the bleb, but it runs through pretty much, I would say, more than 3/4 of the bleb. And I think a lot of that bleb stability is because of microtubules, because that's very, very close to the plasma membrane. So absolutely have microtubules there, which again is something to look at future, have not explored.
Okay, great. And the second question from Nagaraj is, have you looked at cholesterol distribution in the bleb?
Oh, really fascinating question. That's another thing which I want to look at more nanoscale. Quick thing, yes, I do see a lot of this sort of different spatial differences in cholesterol distribution, but also other lipid order markers, which I didn't talk about. That's another kind of project on its own, I feel. So because of the segregation of the transmembrane protein receptors and other kind of proteins, I think the sort of the lipid environment at the back versus the tip is very, very different. So I do see a lot of cholesterol actually at the back. But again, like how is that happening and all that is completely unanswered in my case, at least.
Okay, fantastic.
Really yeah.
We have a couple more from YouTube, from Purnati Kuntia. Does the Picket formation lead to selection for which transient bleb will become the main bleb, or does it form after the bleb selection?
Sorry, if the Picket sort of gradient is formed?
Does the Picket formation help select which of the transient blebs is going to become the main bleb, or is it forming after the main bleb has been sort of chosen?
So that's a really interesting question, because the way at least I see when these blebs are forming is that the gradient for CD44, even the CD44 itself has a gradient, and that's very fast and it's very sharp. So the way we think this whole Picket mechanism is really working is, let's say you are throwing out a lot of these blebs, there is this timing which it controls for myosin. So eventually all this is leading to myosin activation at the back of the bleb. And the more myosin you activate, sort of the more stable the bleb is. So I think initially it's a very stochastic process. However, there is one bleb which goes out sort of really, becomes a really far bleb, and the timing for that picketing mechanism is like sort of instant and quick enough for it to make more myosin at the back of the bleb, and that sort of selects for a bigger bleb or a more stable bleb. But this is in terms of the temporal scale is not really worked out. So I have only worked out trying to figure out what are the components, but the temporal regulation is something which I haven't really addressed yet. Yeah.
Okay, fantastic. And then more questions about the transient blebs. So from Carrie Remisia saying, are the EGFR, CD44, and RAC1 involved in regulation of the transient blebs?
Yes, so I think it's true, especially for CD44. So if you go back to this really beautiful work done by Victoria Sansperino lab, where they're looking at actually CD44 in this melanoma system, you can actually see if CD44 is removed, cells tend to sort of switch more towards lamellipodia-like migration. And that's very fascinating. Again, temporally, it's not done, but it kind of indicates that even for those tiny blebs, which in their system they're talking about, it's really something which is important for amoeboid migration. This is thought to be done via also myosin regulation. So what Victoria's lab is suggesting that it regulates myosin activation, which I think is even important in my system as well.
Okay, great. And then a follow-up question. Do PIP2 and PIP3 also form gradients in the blood?
Yes, so I didn't show it at all, but so PIP2 is hard to know for gradients because usually the plasma membrane is like 70% to 80% PIP2. So the local changes are very hard to judge, but PIP3 is easy to check. And PIP3 sort of follows the same gradient pattern as RAC. So that's again sort of follows the whole trend of EGFR signaling, PIP3 and RAC at the back of the bleb. Absolutely.
Awesome. Thanks so much. Okay. Thank you to everyone who's on YouTube for asking all those great questions. Returning the questions. Yeah. Returning to the questions in the chat here. Two from Ernest Liu. First, does knocking down CD44 change the expression of the EGFR or the amount of the EGFR on the plasma membrane?
So those are two really, really good questions, which I think I should address. So I'm going to keep that in mind. I don't know. I haven't done like fluorescence correlation spectroscopy for like the amount of EGFR on the plasma membrane, total amount. I don't know. But those are really, really great questions. And okay, great. I see his other question too about inhibiting formins. Again, I have not done that, but Matthew Peel has done that, and it does impact the bleb stability. So they do have a paper on bioarchive about the whole regulation of actin sort of distribution and how that impacts bleb stability. So they do inhibit overall all formins and check that. So it is important for sure. We just don't know which formin is important.
Okay, fantastic. From Arabinda, do the blebs retract in the migrating cells?
Yeah, they do retract at some point. So they do retract, but it's usually very stable for at least an hour or so. And the way they retract is, again, how we classically think about bleb retraction is, again, you will have the cortex formation all over till the tip. You start getting more and more myosin towards the tip, and everything sort of comes back. So still behaves like a classical bleb that way.
Okay, great. So we have a question from Anurada, and then Leon will ask his question. So any thoughts on whether the membrane organization determines the site of the bleb formation? Yeah, I was also wondering about that initial formation. It's really fascinating.
Yeah, so so that's really interesting in thinking of like the symmetry breaking event itself, right? In any sort of cell migration. And I absolutely think membrane organization really impacts that, because if you think about the place where the symmetry breaking event is going to happen is often where you don't have things like esrin or anything which would make the cortex really strong. And again, like as I said, I do think it's important, but again, at the scale of anything temporally regulating that is something I have not at least looked at. And this is one of my future projects sort of to do, yeah.
Awesome. All right, Leon, please go ahead.
Hey, Leon.
Hi, Ankita. Super nice talk.
Thank you.
Really enjoyed it. It kind of building on the last question is very similar to what I wanted to ask. So what do you think is the role of actually the confinement process? Do you think that kind of sets up the gradients that then gets driven forward and sort of persists?
Yeah. So something which I think confinement is 100% doing is increasing the contractility, overall contractility of the cell, right? So again, going back to work which was done in Matthew Peel's lab and also Vereena's lab, where once the nucleus gets crushed, there is higher CPLA signaling that happens from the nuclear membrane that leads to sort of a spike in the contractility. And once you have that spike, you can imagine now the sort of cortex becomes unstable. So this is again going back to these instabilities we have often thought about in context of sort of a symmetry breaking event, right? So again, it's very spontaneous. You have a spike of contractility. Everything sort of becomes unstable, and that wherever there is sort of a weakening of the cortex is where you will start forming the bleb. And again, as I said, like I think it's a matter of timing then when myosin sort of gets high enough around the neck that leads to sort of cell choosing one stable bleb. And once that is kind of established temporarily, the system is kind of pushing everything towards that one giant bleb, because that's constantly growing, and now you have more and more of this picketing mechanism going on at the back of the bleb that leads to sort of maintain the stability more and more. Because if you think about something like CD44 being removed from the system, you can clearly see that they are still throwing out the bleb, but it's just not able to sort of stabilize it. So I think the importance of this sort of a mechanism is to really control that timing, and also the amount of sort of contractility at the neck. And if that answers sort of your question.
Definitely, yeah, that's great. Just, can I follow up cheekily just a little bit? Could you sort of predict cytoplasmic flows from this? Do you think that there's a flow from the cell body into the bleb that then leads out? Or what happens to the fluid in the bleb?
Yeah, so that's 100% still going on, right? Because the way the bleb is still being pushed is because there is a cytoplasmic flow, which is still going on more towards the tip of the bleb. And I guess at some point there is a backflow of some sort, which I haven't tracked in the system, but is really fascinating. Because also thinking about all the other proteins, which are not transmembrane proteins, I think cytoplasm is going to play such a huge role. So I think when I start addressing questions about GEFs, or everything from cytoplasm, microtubules is going to be more and more impactful, at least understanding the gradients for those proteins. Yeah.
Yeah, and things like G-actin monomer recycling.
Exactly.
Like yeah.
Absolutely. Yeah. There's so many black boxes right now in the system. Yeah.
One big mystery. It keeps us employed, right? So there we go.
100%. Hopefully, fingers crossed. Hi, Nada, I think has a question.
Yeah, Nada, please go ahead.
Yeah, hi, great talk. Thank you for that.
Thank you.
I just quickly wanted to see if you checked what happens to the inverse polarity between the GTPases as the bleb is retracting, because you said as it's formed, it's stable for like an hour and then it retracts usually. So what is happening with the GTPases when it's retracting? I don't know if you've checked for that.
Yeah, so that's a great question. Till now, my fascination has been the bleb stability, but of course you have those movies where you can actually see them when they're retracting. So I can give you a sneak peek. So something which is RAC is very interesting to me when the cells are retracting, because now you have the RAC gradient going more and more towards the tip. And I think the RAC is really important for the actin, which is very close to the cortex. And that's sort of like it becomes more and more uniform. But the bleb has to sort of already grow a little bit short for the RAC to cover it up. But this temporal thing is a little tricky with the sensors, because sensors don't always give you like the 100% of the temporal scale, but that's the best we can do. I think RAC, really goes more towards the tip when the blebs are retracting, if that's answering your question.
Yeah, it does. Thank you.
Thank you so much. That's actually really fascinating to think about. The way we understand an adhesion-based system, we don't really understand that in bleb-based system at all. Aashrith.
Yeah, absolutely. Aashrith, go ahead.
Sure. Yeah. Great talk, Ankita. I was just curious, about the cell volume regulation when you're confining it. Like, is that changing a lot? And that's what it's leading to the instabilities at the cell at the cellular front? I'm just curious about the cell volume regulations. And if you change the confinement, you're basically putting a solid stress on a cell.
Yeah.
Would the amount of solid stress that you put based on the confinement, does that affect the bleb formation too? Like if you change the solid stress based on the amount of aggros you put on it or things like that?
Yeah, those are really great questions. And I'm thinking about the overall volume. I mean, it does get flattened out, but it also sort of gets squished. So I don't have a sort of a right away answer for you, how much is the volume change, because that's something I have looked at, and I don't know anybody who has really measured it yet. Maybe it's somebody who is doing it, but coming to this question about the stiffness, like the sort of the concentration of agarose or something you would put, I think it absolutely has an impact. Like you have to control that. Again, I don't know whether, like how much change in the stiffness is really important, like what kind of mechanical, all those questions in terms of mechanics is something I don't think the field has at least addressed yet, or I have looked at. So those are fascinating questions, but sorry, I have no answer for that right now.
We do have one more from YouTube.
YouTube is active
asking
today.
I know, it's great. Asking if you looked at ER-PM contact site markers, for example, eSite2, STIM1.
That's a great question. So I know why this question is being asked is because of the calcium regulation. So to give a context, in the smaller blebs, there is a really beautiful paper about contact between the ER and the plasma membrane during the bleb formation and retraction. So you get like a spike of calcium when the bleb is retracting. Coming to these giant ones, I know that there is ER sort of running through most part of the bleb. Again, like it's like microtubule. It almost goes to the tip, but doesn't really go to the very much tip of the bleb here. But I have not looked at ER and plasma membrane contact like STIM or ICE system. However, I know that there are calcium spikes in this as well. So once you crush it, the back of the cell actually has these like calcium waves which goes up and down. I think it has an impact on, again, myosin regulation at the back of the bleb. But again, the pathway is not worked out. But it's a really, really fascinating thing to think about, since we know in those smaller blebs that it's really important.
Great. And from Johan Zhao, does the migration require any chemoattractant? Is it directional or random?
So in the system which I talked about, there is no sort of directional chemoattractant cue here. So they are very persistent, but they are not very directional in their migration. However, there was an experiment which was done by Verena Rupert in her paper when she is crushing zebrafish progenitor cells. That one is really fascinating because she is doing a LPA chemoattractant under sort of confinement for those, under confinement. And what she sees is actually the cells run opposite. So they're not running towards the chemoattractant, but in the opposite fashion. But now thinking about how the receptors are distributed, there are way more receptors at the back and towards the neck. So actually the cell now is contracting more at the back and is pushing in the opposite fashion, and you have your chemoattractant towards the cell body. So everything which you think about adhesion-based system, everything else is going on sort of reverse and bleb-based migration system under confinement.
Awesome. All right, a question from Mike Bischoff.
Yeah, hello. First off, that was an amazing talk.
Thank you.
I tried to wrap my head around like how the retrograde flow is working here, because you have these nicely visible actin bundles which seem more perpendicular to the direction of retrograde flow. So might be a bit of a stupid question, but I try to imagine that. And does myosin contractility plays their role too, or is it only polymerization based? What do you think there?
Yeah, no, it's a really fascinating thing to think about, right? Because there is so much, such fast actin retrograde flow going on, and there's clearly active polymerization going on at the tip. So this is something which was worked out in Matthew Peel's lab a little bit, where if you look at the polymerization rates going from the tip to the back, there is definitely way more polymerization of actin going on at the tip. And what the back sort of gives you is also more crushing of crushing of actin, because there is way more myosin. So again, thinking about like the systems from Karen, where like this retrograde flow is sort of maintained because you have myosin at the back constantly sort of crushing. And again, there is work from Jeremy's lab where he shows that there is more cofilin activity also at the back of the bleb, like these giant blebs. So that leads to more depolymerization at the back. So the rate of polymerization is high, but it still sort of maintains throughout the bleb. However, the rate of depolymerization is much higher at the back of the bleb because of myosin, cofilin, and all those factors being present. Does that answer your question, Mike?
Yeah, I still try to wrap my head around it, but thank you very much.
Thank you. I would really suggest you to see Matthew Peel's paper from 2015. At least it has indication about like how this could be sort of maintained throughout. But like regulation, like precise regulation of all those like actin regulators, we are only beginning to understand. And I think Jeremy's lab is really focusing on that.
Okay, thank you very much.
Thank you. That's a wonderful question.
All right. Thank you so much.
So cool. So many
I think
questions.
we're a bit beyond time.
I know.
So with that, if anyone has any burning questions, feel free to jump in and ask. Otherwise, I will sort of do the official sign off. So see you all next week, same time, same place.